Welcome to Tiny Show and Tell Us, the bonus series where you write in with your favorite science news or factoid, we read your email aloud, and then we dive deeper. I'm Sam Jones, and I'm here with my co-host, Deboki Chakravarti.
Hi, Sam. Last time, we talked about camel antibodies, and we talked about art history debates. So a reminder for all of you listeners, if you have a story you'd like for us to cover on Tiny Show and Tell Us, all you have to do is write an email. So you can email us at tinymatters@acs.org, or you can fill out the form linked in the episode description. Okay, Sam, I'm gonna go first. I have a message from listener Catherine who wrote, I have a genetic condition called Turner syndrome. This occurs where X chromosomes are damaged or missing. I'd love to hear about how this genetic condition was first diagnosed or how research into genetic conditions first started. Catherine, this was a really fascinating history to dive into. And I'm going to start for people who aren't familiar. Turner syndrome, like Catherine said, is when one of the X chromosomes is missing or damaged. And it can lead to a number of different symptoms, including like being short, having heart defects. It can also contribute to infertility. And so we'll get into some of those symptoms in a bit because they're actually part of the history of how Turner syndrome was kind of understood. Part of why I found this really fascinating is I think it gets to that underlying issue of how do you trace some set of symptoms to their cause, particularly a chromosomal one. So Turner syndrome is named for Henry Hubert Turner, who was a doctor who started writing in the 1930s about a group of patients he was seeing that had this like similar set of symptoms. So they had shorter necks. They were just also shorter in general. They also tended to not have gone through sexual development. So he thought that they had something called Klippel-Feil syndrome. I'm not quite sure if that's how you say it, Klippel-Feil syndrome. This is a bone disorder that can happen when the bones in your neck don't separate during like fetal development. So that kind of, might explain why someone has a shorter neck, but it didn't really quite fit the patients that he was seeing because the thing that happens with that Klippel-Feil syndrome is that shorter neck is happening due to a fusion of the vertebrae, and that's not something he was observing in these patients. So you have this physical set of symptoms, and you have this one possible syndrome that can explain it, but the actual mechanism doesn't line up with what he's saying. But what Turner later realized is that these patients didn't actually have a shorter neck. They had webbed skin that was making their neck look shorter, but the bones themselves were actually fine. And when he did different blood tests and urine tests, there weren't any issues coming up. But he was able to find that there was one treatment that was helping, which was injecting gonadotropic hormones. And that helps because patients with Turner syndrome, they have a lack of these hormones, which are really important for puberty, sexual development, and egg release. So Turner kept working on understanding the symptoms his patients were experiencing. And in 1950, so he's still been working on this for a while, he identified more symptoms like a narrower aorta and different eye disorders. And this is also when he wrote his suspicion down that the syndrome was due to some kind of congenital issue, meaning it was something that originated before birth, which is, I think, kind of like where you start tracing that there's something going on that's like happening during the development, basically. Like you're saying it's not just some like kind of later on disorder, like there must be something that happens before birth. And, one of the things that could be is a chromosomal issue. And in 1959, it was actually another research team who was able to trace Turner syndrome to a missing X chromosome. And Turner confirmed this in 1962. And one of the things that was interesting to me about this is that work was based on twins who had Turner syndrome. And one of the things he found is that these twins actually had something called mosaic Turner syndrome. So this is where not all of their cells are actually missing the X chromosome. So in their case, their white blood cells actually had both X chromosomes. And so people with mosaic Turner syndrome can have less severe symptoms compared to people whose Turner syndrome is like all of their cells are missing that chromosome. Yeah. He's like really the person, I guess, who kind of informed a lot of our understanding early on about Turner syndrome, but people are still working on it, including figuring out like what conditions people might be more predisposed to and what we can do to like better support them. So yeah, it's this ongoing field.
Wow, that is really fascinating. And just the history of that. And I was kind of thinking about, as you were talking about this and mentioning dates and years that this research was happening, Like the fact that DNA, the double helix structure of DNA was discovered in 1953?
Right, yeah.
The idea of just thinking about genetics and heritability and like, yes, this is something that for decades at this point, people have been saying, like, there's something that's going on. Things are heritable. Like we're seeing things in parents that we're seeing in, but really being able to like visualize what could be going on.
Yep.
That was such a recent thing at that point.
Yeah.
So it's kind of amazing that they were able to even do what they did in some ways
absolutely.
back then.
Yeah, I was kind of like a little surprised that it was like 1959 that they were able to narrow in on this is actually like a chromosomal issue.
Yeah.
I just think that was like so like, I mean, it's so important, right? Like it's so important to be able to do that visualization and actually see like, there's something here that explains what people are experiencing.
Yeah, absolutely. American Medieval is a podcast about the Middle Ages, but with an American twist. Every week, Professor Matthew Gabriel is joined by an expert scholar to talk about either some bit of the medieval world itself or how Americans have, throughout our history, used the Middle Ages to say something about ourselves. If America has never been fully modern, it might be because we've always been a little bit medieval. New episodes are available every Wednesday. So subscribe to American Medieval in your podcast app now. That's American Medieval from the Multitude Podcast Collective. Well, Deboki, I have a tiny show and tell us for you today, but first I have addressing a listener inquiry question. I don't want to say concern, but okay. So this is from listener Oliver, who wrote in saying, "Hey there, just a note on a previous episode, the Making Labs Safer episode. As always, a very interesting and good episode. I tuned in at episode 3 back then and am subscribed to your podcast ever since," which is, wow, that's amazing. Thank you, Oliver.
Yes.
And then Oliver says, "But, oh no, here it comes. I wish you'd also explained why the 2 example chemicals mentioned are so toxic, how they get absorbed, and what mechanisms are involved so they can and do wreak havoc in our bodies. No lengthy explanation, just a small breakdown. Maybe something to consider in the future? Kind regards, Oliver." And so I wanted to include this today in this episode just because Oliver wrote in a while back, like we often say, like we have a stack of listener stories and questions and whatnot all the time that we're going through. But this was a good point. So for anyone who's listening that is like, what is the Making Labs Safer episode? We talked about 2 lab accidents. One of them with dimethylmercury, a chemist at Dartmouth named Karen Wetterhahn died many months after being exposed. And then the other one was a prion exposure. I believe she was either early grad school or lab tech at that point in a prion lab, poked herself, was exposed to prions, and then about a decade after her exposure died of neurodegeneration, which is horrific. Both were horrific.
Yeah.
But I just want to really briefly touch on dimethylmercury and prions and what we know and what we don't know. This is very condensed, but I wanted to address Oliver's note because we love when people write into us and are like, I want to know more about this thing. What the heck?
Yeah.
We listen. Even if it takes us months to get to it, we will listen. Okay, so dimethylmercury, how it gets absorbed. So it's super lipophilic, which means it can dissolve into the lipid-rich fatty matrix of your skin. And from there can be absorbed right into the bloodstream. So that is not great. And the reason it's so dangerous is that it'll quickly become metabolized into monomethylmercury, sometimes just called methylmercury. And methylmercury is attracted to sulfhydryl or thiol groups that are on proteins. And these groups are crucial functional groups that not only dictate the stability and the structure of a protein, but also their activity. And so all of a sudden, these proteins throughout your body are under attack. You're getting mitochondrial damage. You're getting proteins aren't being made. You've got oxidative stress going on. Like it's just a complete mess.
Yeah.
And so that's the basic mechanism behind why dimethylmercury is so dangerous. And ultimately that will lead to neurological decline. Like it's shockingly fast, actually. It's not like an instantaneous thing, but the fact that Karen Wetterhahn died about a year after her exposure to like a minuscule drop of dimethylmercury just like shows how dangerous this can be.
Yeah.
So prions. Prions, so these are misfolded infectious proteins, the ones that cause things like mad cow, right? And the way they reach the brain is typically by accumulating in lymphoid tissue. So the spleen and lymph nodes. So you eat something that maybe has them in it. In the case of mad cow, it was burgers, but you also have other diseases that essentially if you eat something where there are prions. Typically, that is brain. You get like a mix of that, or it's central nervous system tissue that has these prions in it that somehow gets into your food. So you could be exposed to prions, and that's really bad. So then from there, they will slowly, remember this can take a decade or more potentially to show up. They will hijack peripheral nerves and then move into your central nervous system. And the way that they seem to do this is through exosomes, which are these extracellular vesicles that bring stuff in and out of your cells, or tunneling nanotubes, which are these transient membrane channels between cells. Compared to something like dimethylmercury, the mechanisms behind how prions do what they do is way less well understood. Like really understanding the mechanism of how they do what they do is something that a lot of labs are studying.
Right.
But these pathogenic prions, they will bind to normal cell proteins, causing them to misfold. And then you get this chain reaction of misfolding. And then they aggregate into plaques and fibrils that then further push that chain reaction along. So, Oliver, I hope that helps answer your question. It gives you a little more info on mechanism. Again, there's a lot more that I could say, especially about the dimethylmercury, but I think that covers it succinctly enough.
Yeah.
So yeah, thank you, Oliver, for writing in.
Yep. Thanks for your questions.
So now I'm going to talk about a Tiny Show and Tell Us from listener Hannah. Hannah wrote, scientists engineered yeast to provide the same nutrients as pollen to give honeybees a balanced diet, leading to a significant increase in reproduction and colony size. I love it. I love ending on like a happy, this is like great.
Yeah.
Science can be flawed, yes, but it makes so many incredible things possible. And I love we're helping bees.
Yeah.
Let's get into it. Because of climate change, honeybees don't have access to all the floral diversity that they need for nutrition. And so beekeepers have started feeding them artificial pollen substitutes that are made up of protein, flour, and sugars. But the issue is that these substitutes don't have the right sterols. So this is a subcategory of steroid alcohols. Mm-hmm. They're fats, aka lipids. The bees need them. Like nutritionally, the bees need them.
Right.
And as I started reading about this, I immediately thought of baby formula.
Right.
Like how we talk a lot about like, okay, we have breast milk, you have baby formula. We're trying to recreate what's in breast milk and put it in formula, thinking about, okay, what fats are present, what other things. Like I think of this being like bee formula.
Yeah.
Okay, so this project was a collaboration between the University of Greenwich and the Technical University of Denmark. And their goal was to engineer the yeast Yaroia lipolytica, perfect, lipolytica, love it, guys, to make 6 sterols that the bees need. So they picked this yeast because it has a really high lipid content and it's known to be food safe. And it's also used in aquaculture feeds. I actually saw like it's used in tons of stuff. Mm-hmm. And so the idea is to engineer these yeasts, grow them up in bioreactors, harvest them, and then dry them into a powder that the bees can consume. Easy.
Yeah.
But of course, the scientists needed to figure out which sterols the bees needed for their nutrition. So what they did was they sampled tissue from pupae and adult bees to figure it out. Mm-hmm. And based on their work, they were able to engineer yeast that could make those sterols. And then they turned the yeast into bee food and fed colonies. And then they looked at, okay, how did the bees who were fed this yeast versus the ones that were not, how did they fare? How did that compare?
Right.
And the results were pretty striking. So the ones that were fed the yeast had 15 times more viable pupal stage bees compared to colonies on a controlled diet. Mm-hmm. The colonies with yeast also were able to rear their brood for longer. And the scientists found that the larvae in the yeast had a sterile profile that matched what you'd find in colonies that were naturally foraging for their food. So that did suggest that they were getting what they needed. They were getting the sterols what they needed, and they were getting them to their young.
Yeah.
There's still more that needs to be done in terms of thinking about whether this can work at a larger scale. Like it sounds like this is not something that's being mass produced. And also, of course, you want to ask questions about like, could this potentially have any negative side effects?
Right.
Could there be issues for other colonies or the colony as a whole or, pollination?
Yeah.
But ideally, it would only be good things and farmers one day could use these yeast supplements and really help with the success of bees.
Yeah. I like the idea of yeast supplements for bees.
I know. It's kind of like, it's kind of cute. I say that where I'm like, we did this to them, but
Yeah.
How quaint.
Like we created the problem and then we're like, oh, that's adorable.
Let's come up with a cute solution. Yeah. No, but it's very cool. I love the inventiveness of that.
Right.
Yeah.
Yeah. We got to feed the bees.
Yeah.
Well, thank you to Catherine, Oliver, and Hannah for submitting to Tiny Show and Tell Us, a bonus series from Tiny Matters, created by the American Chemical Society and produced by Multitude.
You can send us an email to be featured in a future Tiny Show and Tell Us episode at tinymatters@acs.org, or you can fill out the form that's linked in the episode description. We'll see you next time.